No worries. Okay, so I'm happy to introduce you to Kristen Cruz. She's a teacher at Fullerton High. Maybe you could give me your background, but she's been with the Amgen program for quite a while, and she's had students who've then come to Fullerton College, who then moved on to do great things. And now we said it was a full circle. Your class is going to go visit his research lab, so it's pretty nice to hear that story. But Kristen, I'll let you.
Sure. I work at Fullerton High School. I've been doing this for at least 15 years, the Amgen program, which is really awesome. And so what I thought would maybe be most helpful to you, having not really implemented it yet in high school, is showing you what I do leading up to doing the labs to help make sure students actually understand the content. So I run this with my AP biology class, and if you teach, how many of you guys teach AP? Okay, so with AP, I don't do all the stuff that I'm showing you because we just don't have the time, right? So everything I'm going over here with AP Bio, it's okay, we condense it, we go really, really fast. I also do this with my anatomy class. So I teach 3 anatomy classes, so that's primarily what I do the sequence in, and I tie it in through when we talk about the endocrine system and we talk about insulin, and so that's how I get it to fit in, where we talk about purifying the RFP protein that you're doing is the same basic thing that you use to manufacture insulin on a wide scale. So I also have a regular biology class, and I just do the very first one with them, the doggy drool or the paternity, just the intro lab with them. So I do it in all of my classes, but various pacing, and I don't purify my protein with my AP Bio class because there just isn't time for that part, but with my anatomy, I do take it all the way through. So I just set up for you guys, and if you are interested in the presentation, I have a QR code that is right here, so you can just grab one. I'm sure it's probably going to be linked somewhere for you too, but they're all on the front tables here, so you can grab one of those on your way out. So I titled this just day 1 and so on. Every day when I run these experiments or when I run the lessons leading up to it with my class, I always share them. What are you learning? Why are you learning it? How do you know you've learned it? All of that kind of overview. So this is just what I would present to my students each day. So on our day 1, I always too, just to get my students talking to one another, I have them answer some sort of question. And so the reason why I put this one on here is because this is for my anatomy class. So we talk about the history of insulin production. So that's why that particular slide is on here. And then I actually have not really found a history of insulin write-up that I liked that well until that chat GPT or whatever that new AI thing is. So I actually had AI write this for me, which it was better than anything that I found. I found stuff that was either too high level or too low level. So I was like, hey, I want you to talk about this and this and this. And that's what AI did for me. So I have my anatomy classes just kind of start off with a little, hey, what is the history of insulin? And do any of you guys use Pear Deck in your classroom? Okay, I only have a couple of you. So if you use Pear Deck, then these slides are Pear Deck slides. If you don't use Pear Deck and you're like, what is Pear Deck? I don't know because our district buys it, so I don't know what the free version does, but Pear Deck's really cool because it's a way for students to interact with you and you can read what they're writing. Like for instance, right here, I'm having them analyze diagrams before we get into it so they can figure stuff out. So I asked them, hey, what is a plasmid? How does it look different? And before we talk about any of this stuff, they're doing diagram analysis. So if you don't use Pear Deck and you like this, then just delete the bottom there, and however you run it in your classroom, you can. And if you do use Pear Deck, then you can use, this is already set to go for you. So the first day, it's basically, we do the background reading, what is insulin. I have them go through, the cool thing about Pear Deck is they can draw on stuff. So I have them like, hey, look at, here's the plasmid, here's the main DNA. They go through, they circle that. I have them take some notes down. I'm clicking through really fast because there's like 8 days worth of lead up here, and they describe what they think conjugation looks like before they know any of the scientific terms for it. We talk about that. We add some more to our notes here. We talk about what transformation is, or why do they think that wouldn't really happen with high efficiency in nature, and then we talk about how cloning works. Again, more diagram analysis. We go back to that human insulin gene, talk about different transgenic organisms, and the fish that a lot of them have seen in the pet store, the glowfish. So we talk about the glowfish, talk about this lab, and that's pretty much day one. So there is kind of a lot we go through there. My classes are about 53 minutes. So you have all of that with the QR code if you're interested. Any questions for me about what I do on day one with my anatomy classes? Okay, so that's how I just kind of like set the stage for them. The second day, what we do is there's an online simulation. It's really, really cool, and there is a link to it. So it's kind of annoying because the sound's really annoying. Have you guys seen, anybody seen this?
Yes. Yeah.
Okay, so I use, if you haven't, I'll just click on it really quick so that if you haven't, you can see it. It's,
You're not joking.
yeah, it's really loud too, but it goes through how gel electrophoresis works. It's from Learn Genetics, the Utah site that's really, really cool. It's kind of a little bit, it was made a while ago, so it's kind of long and there's a lot of clicking through, but I think that it helps students really to understand how this process works. And then at the end of the period, they can usually get through all of it. And then I have them, there's just like a really short quiz with some of the general, like, hey, how does this stuff work? So they just have to like answer a quick question for me about DNA and how it moves through the gel, and be able to really quickly defend a claim about the size of the fragments in 2 of the different wells. That's just like how they show me. I tell them, I'm gonna go through this, and at the end of the period, there's gonna be a really short quiz where if you did it, you should be able to do well on the short quiz. So that's my day 2 leading up into how electrophoresis works. Any questions for me on that one? All right, so this, and with my AP Bio, I don't have time to do that. I could assign it for homework. I also have them, a lot of my students that I have for AP Bio have had, because half of my class had me for anatomy, so they've done this. And then the other half, I can just tell them the important parts of it, and they're gonna pick it up. I can assign them like a short video to watch before we do that on how it works, so that I am not spending that time, because you know how packed our AP schedules are. Okay, day number 3, and I'm talking fast to get through all of this stuff, so let me know if I'm going too fast, too slow. Day 3, this is where I teach them how to do micropipetting. We can practice pouring gels, we practice loading gels, and I did a different version of the micropipette art that you guys might have done in here. So, going over how to use the instruments, and mine says H instead of P because that's the ones that I have. So if you're going to use something like this, I printed this out on pieces of paper and I laminated it instead of using the little plates that you guys might have. So I used this year and it worked really well. And then I also gave the kids a challenge where if they finished early, and because of the time crunch, we did not get here. We also have something called a student support period though, which is, it's annoying and rotates, and it just it makes every week we have a different schedule. And then sometimes it's attached 25 minutes to this period or this period. So this is something I would go back to, having them do their own art during the study hall session when kids don't have work to do. So I thought I put it on here. Maybe I did not. Dang it. I thought I gave you a picture of it. Let me see. Sorry, I'm gonna exit out of here and see if I have the right one. This one? Oh, there we go. Okay, so that's what they ended up creating. And yeah, I made a rainbow and I made it with different sizes so that the little clouds at the bottom were larger. And I had kids able to like just barely finish by the end of the period. I also though have this down because now I'm doing it for 15 years so we can like move like this. So it probably won't go that fast for you the first time you do all of these things. So in that period we practice how to load. I take gels that I just like remelt and re-pour and I like cut them so they're like, you get 4 little tiny wells in this gel to practice with, and then we go into this. All right, any questions there? I also had, I had a student who wrote out a procedure for making a heart that was really cute also. Cool. Okay, so here is the next thing that I do, and I do this with my anatomy class, and again, to tie it back into anatomy, since we've been talking about diabetes and insulin and endocrine system. So with my anatomy class, I use the doggy drool or the cat paternity stuff, and instead of using that write-up, I use that write-up for my freshmen, but instead of using that write-up, I use those samples, and I instead have them do something where they are running a gel electrophoresis, but they read about type 2 diabetes, and I looked at several different genes that I found information of the size of base pairs in a mutation, and so this is fabricated, but kind of accurate-ish. So I found just a couple genes here, and there's more now than there was when I first wrote this. So I say that we're testing for these specific gene mutations. Kids will run the gel, and they know their reading that I just had in here that I'm leaving out goes and talks about some background and the combination between genetics and environment. And here they have the size of the different mutations, their base pairs, and then, I think I put a bend here. Here's a bad blurry picture. I got way better results, and of course, the one that I chose to put in here is the not great result, but it also doesn't look so great on the screen. It looks better on my computer here. So here are the results, and then what I have students do is they go ahead and they make posters. So one day they run the electrophoresis, and as we're waiting for it to run, I have mini ones, so as we're waiting for it to run, then we go back and we do the reading. So I tell them, like, you're acting like a lab tech working at wherever, and you are running these samples. And then today, now you are a genetic counselor. This is what you are going to be doing. So here is what I have them do. They have to address this question here: what mutations do each of the patients have, and what does that mean for their susceptibility for developing type 2 diabetes? Some of my kids did extra research and found out that I think it's like this gene mutation is more strongly associated with developing type 2 diabetes than some of the others. They talk about how it's lifestyle and environment and genetics. So they answer several questions, and then they use this information to help them make their claim. Okay, so again, that's going pretty fast. These are works in progress. So they had about a period and a half to work on this. So if you're going back and reading these, none of these are awesome yet, but I did this this week. And so I took a picture here for you to see works in progress. Any questions about that? Yeah.
Sorry, are we looking at, is that your
These
anatomy
are-
class?
This
or
is what
PDI?
my kids did like on Thursday.
No, which class?
My anatomy.
Okay.
Yeah, so this is all what I do in anatomy. For AP Bio, it's pretty much like, hey, we talk about how this works, you watch some videos for homework, we run it, we analyze the results, we move on.
And you mentioned you do something similar for your regular bio?
For regular bio, I would do the same, like the poster, but we don't do insulin, we do the actual, the lab, the paternity lab,
Got
or
it.
the, yeah.
Thank
So, that way when I see them maybe 3 times over 3 different courses, they won't have done the same things.
Oh, nice.
Yeah. And any other questions? Okay. Wow, this is going really quickly then. Cool. This is a super fantastic resource. I love this resource. So this is a, okay, I must have popped on right there when I wrote homework. I usually don't give homework in my anatomy class. That's probably what I had my AP Bio students look at. But if you click on the link there, it takes you to this E. coli insulin factory, and so you build a paper recombinant plasmid because students just really struggle with all of the like little microscopic stuff that's happening and keeping it all together. There's like so much terminology. So, I'm just trying to scroll like it's my interactive whiteboard here. So everything that you need, all the background information is here. There's a sheet where kids write about restriction enzymes. Here is plasmid DNA. Here is human insulin gene. Here is restriction enzymes and where they cut. So you can go ahead and use this material because it's a really awesome lab. Before I have them do that hands-on lab, this is a really neat example of like what it looks like, a computer animation showing it more like in nature what happens. This is a really quick video. I think it's 2 minutes. It's really a good resource. This is what the paper plasmid lab looks like when the kids are going through it. So they have to go through it. They have to figure out which restriction enzymes they should use to build their recombinant plasmid to be able to insert that human insulin gene. And so they mark up on the human insulin gene, they mark up where they have to cut on the plasmid, and it's cool because there's a couple different ones that will work. So they have to figure out which one is gonna make the cuts closest to the gene, and it's really easy for me to see if they've done it correctly because it makes like a necklace, so I can put the necklace on, and then if they didn't choose the right one, it's a whole lot longer. And so it's really easy, like, oh, there's a better one, go back and try again. So I was able to get them to go through this in about a lab period and finish it. This is what it looks like when they complete it. So it's just like the loop of the plasmid there in red, and then the white gene, and it helps them to really understand that process. Yeah.
Did you make that or did you purchase that from the company?
No, that's that link that I gave you. Everything is free.
Okay.
So to save time, you have the students assemble it, but to save time, I just glued them all, and it was annoying because I was like, okay, for like knowing I was gonna do this, like, all right, I have 5 free minutes, let me start gluing these things together because we don't have TAs. So yeah, everything's totally free. Yeah, it's awesome resource. Okay, let's see, what is this thing that I'm doing? Oh, here I've included this for you. I made these sketch notes. So if you print this out, this kind of goes over the process that we're doing. So I fill it in. This is how I filled it in. So this is pictures how I filled it in. So you can go back and you can look over those. If you like to do notes like that, you can photocopy that handout, give it to your students, and then do notes with them that way to explain what's the recombinant plasmid that we're making, what are the features of it, what happens when you get it inside of the E. coli bacteria. I do some other notes later on also to help them with transformation, but again, all this stuff you guys have access to on that QR code. All right, any questions on this one? And I do this with my AP Bio kids too. So, like, this is like, they really need to understand this and they like struggle a lot and it's hard sometimes, yeah.
Question on your students that you're getting,
Uh-huh.
they've all had, have they all had, high school bio, or some of them are freshmen?
No, when I do this, our anatomy kids are juniors and seniors, but it's also not a very high level necessarily group. So my anatomy is like a general level. It's not like the honors kids. Sometimes they're in there, sometimes they're not. My AP bio is juniors and seniors also. So our school, the way it works, I wouldn't have any freshmen.
Okay, because I would struggle like doing some of those early labs, like day 3, that
Yeah.
sounds like a nightmare.
Yeah.
I have students who've never had like a bio class.
Yeah, so then you would have to
I'd have
like
to kinda ease
supplement
them in a
it.
little bit.
Yeah, so mine are all juniors and seniors and they at least should have some background, but some of them are the ones that are coming in from COVID years when they had bio, so
Yeah,
they're
that'd be
still a
great.
little weak, but there's a lot of the support with like the analysis that they do with like look at the diagram, let's analyze these things. So it's scaffolded enough that they're, even if they don't remember a lot, they've been okay.
Do the other teachers at your school do this as well?
I'm the only teacher.
You're the only
So,
teacher.
but I'm the only anatomy teacher and I'm the only AP bio teacher.
But the general bio, do they
Um,
do what you do? I'm just curious if you have any coordination.
She does, they do just the electrophoresis, gel electrophoresis. But they haven't gone through they haven't ever gone to the workshops here. And in our general bio, we don't do the full sequence anyways.
What about the other Fullerton Unified School District?
We don't really articulate with them. So I know that one of my friends that teaches at one of the other schools does. I don't know, Joe, besides Sonora, does anybody?
Yeah, we have Sunny Hills.
Oh, okay.
Does anyone do this?
What about Troy?
In Troy? Well, Troy
We
is
don't,
STEM
we don't
school.
do Troy, we
Yeah.
don't have it
They're
sponsored.
their own school.
But
Yeah.
Sonora. for sure.
Troy, I think,
They have their own
is more
program.
content and less lab.
Okay.
So I think they're more like college lecture
Right.
in a lot of their classes, not really as hands-on, I think, but.
But if they were willing to collaborate, you guys would make sure they all do the same labs? Like Sunny Hill, you say Sunny Hill?
I mean,
does it?
we don't, that's just not how our district works. So, like, I don't know what goes on at any other schools, we're kind of like, hey,
So,
we're
and it
here.
depends on the year, cause,
Yeah.
I used to help 3 teachers at Fullerton High School and 3 teachers at Sunny Hills, and this year, only 1 from each school. And
Yeah.
Sonora is only Amy now, so it just depends, yeah.
And I know, I mean, one of them I work with, but that's just because we're friends. So it's not like, that's not how our institution does things. This is also a really awesome video. I think that this is probably made by a college student, but she goes through, and I think it's like a 7-minute video. She models transformation, and I like to show this after my students have built the recombinant plasmid, because then they're like, oh, okay. Even though you talked about it, these concepts are challenging, and so it takes a couple different ways of seeing things for them to be like, oh, that's what's happening. So this is a video, there's a link in it. I usually show this video like when we're actually doing the lab, I think it's lab 4 where you do the electrophoresis. I show this when we're waiting for it to run, the gel run after we've loaded it. Okay. This, if you have some weird schedule like we do, where we have like a student support time or like a study hall or whatever, I made this game a long time ago that I sometimes play if, like I said, we have a weird study hall schedule. So these are just cards. You can photocopy and cut them out in different colors. If you've ever played this game, Happy Salmon, and if you haven't, I included a link so you can watch the video. I took the idea of the game rules and applied it to my own cards. It's just like it can help with the kids like understanding terminology, but it's not necessary. I would only do this like if I have my student support time. I'm like, okay, cool, we got 25 minutes to kill and you guys are all caught up. So there it is for you. I won't talk about that much. You can figure it out based on what I've written here in the sample video of how you play the game probably. Okay. So this is something else that I do. And I did this with my AP Bio class as well. My anatomy class, when I do it with them, it'll take them the whole period. My AP Bio class was able to do it like super quick. So I then have them model gel electrophoresis using their lab tables as like setting it up like an electrophoresis chamber. Oh, here are some of the questions, and I have a link. There was a link somewhere, right there. So there's a link to the handbook. This is the pages from the handbook. Some questions here, again, like to help them analyze what's going on, remind them of the components of the plasmid. But then, this is what I did. So I cut out different, I made paper strips here, so you could photocopy these, cut these out if you want to. I cut them in black and white, and then I take them together to mimic like the big circle and like supercoiled plasmid. And then I have them, we write on our desk, our lab tables, I use chalk markers all the time. So I have them use chalk markers to set up, like turn their table into an electrophoresis well, and they have their micropipetters, and they're like, I am going to pretend to aliquot this sample right here. And they just lay them right on the tabletop. And here is a picture of my tabletop.
Super cool.
Yeah, it worked really well, because they used to struggle with like, oh, now I see this gel, what is it
dry
mean?
run.
It's a dry run, yeah. And so, and I have them like, now we're gonna turn the machine on, and they all
how
like,
cute. It was
pretend. Yeah, they pretend. So, yeah, and these are, like I have like big plastic tubes that I just like, okay, they're just like, okay, let's take it and put it in. So, that's cool. And, I'll put that video on again. And that pretty much takes me with my anatomy class up to, and now we're actually gonna do these labs. That's my lead up. I also, when we actually are doing the transformation, we'll like take more notes, like why is this working, or how does everything work. But this is the lead up so that they understand what can be a really complicated process. Like the first years when I did this, they all were like, oh, this is really cool. And then I said, okay, let's talk about like what's going on. And they were like, oh, well, I missed a lot of that. Like it was fun. I know I made something glow, but they couldn't really explain. So this has helped them to be able to actually articulate what they're doing. So that's pretty much it for what I prepared. You can send me an email. I probably should have put my email on there, but I'm Kristen Cruz, and you can find me if you go to my Fullerton High School website. My email is kcruz. I can just tell. Yeah.
What's that?
Sure.
I think you need to give us access to.
Oh, really?